Welcome to The Explainer. Today, we're diving deep into Echovirus. Now, this is a pathogen that poses a classic and, frankly, fascinating puzzle for clinical microbiology. How can one of the most common viruses we encounter also be responsible for such incredibly rare and devastating diseases? And maybe more importantly, how can our very best diagnostic tools sometimes miss it completely? Just a quick heads up, all the information we're about to go through was compiled to give you a really clear, consolidated view of the current science. And for anyone who wants to dig into the primary literature, all the sources are linked for you. You know, this quote just nails it. It perfectly sets the stage for our entire discussion. It frames Echovirus not just as a virus, but as a clinical puzzle, where what you see on the surface is only a tiny fraction of the real story. So here's our roadmap for today. We're going to start by looking at exactly why Echovirus demands our attention. Then we'll define the virus itself, explore its full clinical impact, and then we'll tackle the really critical diagnostic and therapeutic challenges it throws at us. All right, let's jump right in at the most urgent point, the really severe cases. These represent the visible, dramatic tip of this iceberg, that point where this very common virus becomes uncommonly dangerous. So picture this, a newborn, less than 2 weeks old, presenting with a sepsis-like syndrome. This is, without a doubt, the most devastating presentation of an echovirus infection. We're talking about fulminant, multi-organ failure, severe hepatitis, and coagulopathy. The mortality rate is tragically high, and Echovirus 11 is a particularly notorious culprit here. This single scenario immediately establishes why this virus matters so much. Okay, so now that we've seen the absolute worst-case scenario, let's zoom out a bit and really get a handle on the identity of the virus responsible. What exactly is Echovirus? You know, the virus's name actually tells the whole story of its discovery. Enteric from the gut, cytopathic because of its effect on cells and culture, and human is obvious. But orphan, that's the key. When it was first found, it was a pathogen without a known disease. It was a puzzle right from the very beginning. So, getting into the nitty-gritty, taxonomically, echoviruses are small, non-enveloped RNA viruses. Under an electron microscope, you'd see that classic icosahedral capsid. They belong to the Picornaviridae family. And it's really important to remember that Echovirus is a common name, not a formal taxonomic rank anymore. Most of the serotypes that are clinically significant for us are now classified under the species Enterovirus B. Right, we've seen the severe outcomes. But now, let's explore that much larger, submerged part of the story, the full clinical spectrum, which is where the vast majority of these infections actually lie. And here's the crucial point, the thing that really defines this virus. Over 80% of all echovirus infections are clinically silent, or they just cause a mild, non-specific fever that resolves on its own. This is that massive submerged base of our iceberg. It means for every single severe case we see in the clinic, there are countless others out there that go completely unnoticed. But in that space between the asymptomatic base and the deadly neonatal tip, there's a significant middle ground of pretty severe disease. Echovirus is a leading cause of aseptic meningitis globally, and certain serotypes like E13 and E30 are infamous for causing huge epidemics. It can also lead to encephalitis, myopericarditis, and even those rare, scary polio-like syndromes like acute flaccid myelitis. Okay, this brings us to the absolute heart of the diagnostic challenge. This is where the expertise of the microbiology lab isn't just important, it's absolutely essential. So our primary tool, the absolute workhorse of the lab for diagnosing these infections, is the highly sensitive pan-enterovirus RT-PCR assay. It's fast, it's sensitive, and, it's our gold standard for a very good reason. And the reason this assay works so incredibly well is because it targets the 5' untranslated region of the viral genome. This is a sequence that's remarkably stable across most enterovirus types, and that's the key to its broad utility. It allows us to catch most of them with a single screening test. But here it is, the critical question. What happens when the clinical picture is screaming viral sepsis in a newborn? You run your gold standard pan-enterovirus PCR, and the result comes back negative. What did you miss? And this slide illustrates the pitfall perfectly. It's a reclassification trap. The viruses that we used to call Echovirus 22 and 23, well, they're now classified as human pericoviruses, a completely different genus. And their 5' UTR is so divergent that our standard pan-enterovirus assays will fly right past them. They miss them completely. This creates a huge diagnostic blind spot, which is why a specific pericovirus PCR is absolutely essential in cases of neonatal sepsis. So we figured out how to find the virus, and we know how to avoid that critical diagnostic pitfall. But once we actually have that diagnosis, we come face to face with a stark reality of what we can actually do about it. And this is the hard truth. Despite all our diagnostic precision, we are facing a therapeutic vacuum. Dutch guidelines, just like most international recommendations from the CDC and the WHO, all confirm there is no approved antiviral therapy for echovirus. Management is entirely supportive. It's all about managing symptoms and complications. Now, in severe pediatric cases, especially neonatal sepsis, the off-label use of intravenous immunoglobulin, or IVIG, has been tried, but consistent efficacy just hasn't been established. So our role then becomes providing that rapid diagnosis to guide that supportive care and, just as importantly, to enable good antimicrobial stewardship by stopping unnecessary antibiotics. Now, this lack of options isn't for a lack of trying. I mean, look at this table. Several agents have been investigated. You've got capsid binders like plaquenil and protease inhibitors like rapintrevir that showed early promise but ultimately failed, either because of safety concerns or a lack of efficacy. This whole history just underscores how incredibly difficult it is to develop effective enterovirus antivirals, partly because of the sheer number of serotypes and the high mutation rate of these RNA viruses. So where do we go from here? With no specific treatment and no vaccine on the horizon, the focus has to shift. It pivots to prevention and to the absolutely pivotal ongoing role of the laboratory. As you'd probably expect, control really hinges on fundamental public health measures. Because echovirus is a non-enveloped virus, it's incredibly stable in the environment. And that makes surface disinfection with effective virucidal agents and especially thorough hand hygiene the most critical interventions we have to break those fecal, oral, and respiratory chains of transmission. And this brings everything full circle, right back to our role, which is absolutely central to this whole picture. We provide the rapid diagnosis that lets clinicians stop unnecessary antibiotics, we advise on infection control, and critically, we support public health surveillance by making sure positive samples can be sent for genotyping at reference labs like the RIVM. That's how we help track circulating serotypes and detect outbreaks early. Which leaves us with this one final, provocative question for the entire field. We can find the enemy with incredible speed and precision. We can tell it apart from its mimics. But how do we, as a scientific community, actually develop the weapons to fight it? How do we bridge this huge gap between diagnosis and treatment? We really hope you found this explainer valuable. Please think about subscribing for more deep dives just like this one. And we would absolutely love to hear your thoughts or any questions you might have down in the comments below. Thanks for watching.